So welcome everybody. It's really a great pleasure and an honor to be presenting and introducing the today speaker for the series. I will now share a very short presentation, which I hope is clear for everybody. So we're really very glad and happy to have Dr. Laura Ravasi of KAX Consulting Geneva in Switzerland today with us. She's going to tell us about scaling precision radiotheranostics, smart isotope production with small modular reactors. So we are really entering into a very interesting part of our conversation, which is applications of small modular reactors to very important aspects of everyone's life, which is nuclear medicine. Before entering into that, I'd like to just share with you, for those who are with us for the first time, what we are doing and why this series. So we know that energy is really the golden thread that connects economic growth, increased social equity, and healthy environment. So sustainable development is not possible without sustainable energy. This is what Ban Ki-moon said already in 2014. So we are really going through a global energy system transformation for many reasons that we are all aware of. And this is one of the motivation for this series. So the IEEE second presentation series on nuclear energy has been brought to you by the IEEE Vancouver section, the future direction in IEEE, IEEE SASTEC, the Nuclear and Plasma Sciences Society, the Standard Association, and Planner Positive. This series is intended for a broad audience, not only the engineering and technical community. And so this could really open the conversation also to a much broader audience. And we hope that the people who are attending are part also of the general community with an interest in the energy transition, clean energy, and energy alternatives, of course, including nuclear reactor technology and related topics. So you will have here several links. They are going to be posted on the chat, so don't worry about them. They are going to be quite important because they are linking to the series web pages. So you can see also other type of events which are going to take place from the presentation series, if you are wishing to join the mailing list for future events, and also for link for past presentations, particularly the first series. The next talk that I'd like to point out to you is going to take place the 18th of February, and it will be on plasmas for sustainability. So we really have an eminent speaker talking about that. It's Professor John Verbanker. He is an expert on plasmas. We heard already on the previous talk about how plasma can be used for energy generation, but there are also many other applications of plasma that the speaker is going to talk about. Today's talk. So, Dr. Laura Ravasi is an exceptional scientist. She is a physicist, a physician, and she has really a great experience in interfacing the technology with nuclear medicine, the radiopharmaceutical R&D, and of course, the global commercialization of all those. So she's a bridge between clinical, academic, and industrial experience. She's going really to bring on this platform this incredible experience. And about a presentation, we know how important radiopharmaceuticals are for not only identifying the presence of tumors, through all the various techniques in medical imaging, but they can also be used to cure cancers, taking advantage of their emissions and the fact that they could really be targeted to particular organs. The problem is that in many cases, there are part of the populations that cannot really benefit to that, for that, because of the lack of reactors that could produce such radiopharmaceuticals. Small modular reactors could answer to that and enable decentralized medical isotope production and alongside with low carbon electricity generation, and therefore making this very powerful and important technique in medical imaging more available globally. So I will stop talking and sharing, and I'll invite our speaker to share her presentation. And in the meantime, I would like to remind everyone, please to share your questions either on the chat on Zoom or on the question and answer using the question and answer button that you have at the bottom of the screen. So Laura, the floor is yours. Thank you very much.
Thank you very much, Cinzia, for the nice introduction and for having me over here. I hope you see the presentation full screen. Can you please confirm that is the case?
Yes.
Okay, good. Then I can go ahead and relax because this is the worst part for me. So thank you everybody for attending this introduction to what we can hope for our future so as to optimize production of energy and medical isotopes so as to also treat patients to earlier diagnose some diseases and to live in a more sustainable way. That would be a little bit the talk. And I will be happy to answer any questions afterwards. Now I take you through my presentation, which is moving, so I hope nobody actually feels seasick. And we are going to see how scaling could actually be the way to answer some of the challenging questions on how radioactivity can reach to all patients wherever they are in the world. So as Cinzia has said, we do have some reactors that produce energy, obviously, and some nuclear reactors that also produce radioisotopes. But the point is that we are growing, we are always more and more in all over the world, and the number of patients is also increasing, especially the number of diseases that need to be treated with curative intent because they eventually would, if not cured, they will lead us to death. So we really have to play a role in oncology, not only by earlier diagnosing them or sort of screening and better preventing them, but also by treating them with a radical therapy. And nuclear medicine is one of those that right now has been very, very promising. In the past, I would say 10 years, we have had 2 radioisotope tracers that have been marketed, and that is the first time in 2017 and then in 2021 that we, as FDA and EMA, have the opportunity of treating cancers such as neuroendocrine tumors or prostate cancer with therapies that are radio labeled. In particular, they are radiolabeled with lutetium-177, but that is not the only isotope that can be used to treat cancers or other diseases. Therefore, I will walk you through a lot of possibilities that are awaiting for us in our near future. So this is basically, I would say, due to the advancements in nuclear medicine has always been a diagnostic procedure, and the only, I would say, common factor that all the nuclear medicine devices have is the use of radioisotopes. Most of those are either for positron emission tomography or single photon emission tomography, so they decay by emitting a photon or a single photon or positrons. And in that case, we can actually use for example, technetium-99 metastable or fluorinated 18, F18, so as to label some tracer that we call tracer that we eventually inject into the body, and we follow the path of what we have injected, knowing where physiologically it would biodistribute. And according to what the image we see is, we detect the presence of something that is either good or not good, and therefore we choose what therapy we can offer to our patient. That has been for a long time, and it is always going to be useful. But lately, and by lately I mean in 2017, there was a startup that has thought about using the same target, not with a short-lived isotope, but with a much higher energy and longer-lived isotope, such as lutetium-177, and it has been useful to treat the patient with a tumor that was expressing a specific target. Therefore, the isotope was able to go directly to this cell, and by releasing the energy was going to provoke apoptosis, which means the death of the cell. Now, you may see here in my little image that these are different patients injected with different isotopes, and according to what the distribution is like, we are able to say that we see something good or something bad. So it is very important to know what we are expecting as far as distribution, so as to identify what's good and what's not good. Now, the patients we have here on the image are only a little sample of the tracers that can be injected into patients so as to understand what is really going on. And this is the power of nuclear medicine. We are able to see what is going on inside the patient so as to decide if the patient is going to be eligible for the treatment that follows. So if I am okay with seeing the image of such patient, then I decide, okay, then I will definitely use the same tracer, so the same dotatate, for example, but instead of labeling it with gallium, I will label it with lutetium, so as to go and destroy those uptakes here that actually represent some clusters of cells that express somatostatin receptors, although in the body you do not expect anything of such kind in that situation there. So you know that this patient is actually having a non-normal presence of cells that express that kind of receptor. And if I translate this into a clinical setting, then this patient is indeed bearing with a neuroendocrine tumor. And here you have very bizarre names, but all those names represent the name of a molecule that can be injected and goes directly to a receptor that we specifically tailor or target, so that we know exactly what we are going to look for. And this looking for means we know that if the patient has a lot of those, for example, PSMA, then we know that he actually has a prostate cancer. And so every time it translates into a disease. So according to what we inject, it goes to a certain target, and that target is going to be able to tell us if the patient is holding a tumor of which kind. Now, obviously, right now, we have we don't have the possibility of using nuclear medicine for all the tumors, but in our future, we are hoping to expand the capacity of nuclear medicine so as to treat more patients with this kind of treatment labeled with radioisotopes, because it has very little side effects. And a lot of our patients, or even ourselves, we prefer having a very good quality of life instead of having to go under therapies that reduce our quality of life, such as chemotherapy, for example. Now, you may see that I put out here beta and alpha, because those two are the radioisotopes that are mostly used right now in treating with nuclear medicine. So the beta emitter, beta minus, is actually the lutetium-177, and it is right now the isotope that is used for both treatments that are on the market in US and in Europe, and both for the neuroendocrine tumor treatment and for the prostate cancer treatment. Now, unfortunately, there are some patients that are not responding to such energy from the beta, from the lutetium, and we have clinical trials using alpha. Now, you very well know that alpha emission is more powerful than beta emission, and we actually have a sort of 20 times more effect when we use our alpha emissions onto our cells, for sure. And one candidate that can be used in clinic is, for example, actinium. So actinium-225 is right now in a lot of trials. Some are in preclinical phase, but others are also are already, I would say, in phase 3, which is the phase right before commercialization. And that would actually help treating the patients for which the tumor does not get killed by the beta emission, which means the proliferation of the cells is so fast that you actually do not have enough cells that die so as to overcome the replication of the tumor. So you need something that is going to be even more potent, such as alpha. And not only could it be treated by a lot of doses of alpha, but sometimes with a combination of some injections of beta emission and alpha emissions. Now, I'm saying that this is very new, and it is true that it is very new as far as current therapy, but I would like to share with you a paper from 1993 where it started being possible to use actinium-225 for radioimmunotherapy. And so when I say it's the near future or the very short path, we always have to consider that those studies take quite a long time. So sometimes when I say 10 years, it's almost nothing. 20 years, we start becoming a little familiar, but there is still a lot to learn. And we have to keep very humble in this space, because it's not only how to think of the supply chain and how to manage with the regulatory bodies of the FDA and EMA, it's also how to feel with patients being told that they will be injected with radioactivity and all the radioactive negative burden of the Second World War that a lot of us still have in mind. So I believe that is something that is going to be another challenge for acceptance, although it has a very good potential for clinical purposes. So now I'm going to go a little faster, because what I would like to say is once the patients get at the last resort, which means the standard of care does no longer provide any benefit on their disease, what happens is that they usually feel very sorry, very desperate, very depressed, because obviously there's nothing more that the medical team can propose. And that's where we started. We started with patients who were very sick and with a very miserable quality of life. So daily they were really unhappy. And by treating them with this radioactive treatment, they said that they were feeling like they were born again. They were for once alive. They were able to have a normal life. So we were very impressed by their positivity, and they were our best supporters in going to the regulatory agencies to share how well they felt after being treated, because it was not that much about the tumor shrinking, but it was about being able to live a day without being sick. And that was something that really was important for us. And what we realized is that 22,000 patients have already been treated from on what used to be an orphan disease, which means very, very little number of patients are going to have this disease, with what we were able to have approved in 2018. So we are very proud of the benefit that at least our trial was able to provide to the world. Now, a few years later, we attempted a disease that is not orphan, which is the prostate cancer. And indeed, we went to market with an agent that is again labeled with 177 lutetium. And that was only 4 or 5 years ago. And we already have 35,000 people who have been treated with lutetium 177 PSMA 617, which is what Pluvicto is. Now, both patients from the neuroendocrine tumors and the prostate cancer cannot just be treated because they come and they say, okay, let's see, I have a diagnosis of prostate cancer, so can I be treated? No, we have to make sure that we detect inside the patient the receptors that we are looking for before we inject these treatments. So by enhancing this, I want to support that it's not only the number of treatments that is enhancing, but it's also the number of diagnoses that are made so as to make sure that the patient is eligible for treatment. So there is a tendency of growing on the nuclear medicine field, both on the diagnostic side and the therapeutic side. And right now, as I was saying, we only have 2 isotopes that are approved, and both of them are labeled with lutetium, but there are 2 others in phase 3 with actinium. So the next kid in town that is going to be approved is probably going to be the alpha therapeutic, and that is going to be a very interesting step for the nuclear medicine community. So even more important for the lutetium and for the actinium, we want to make sure that we have a good diagnostic agent that goes and detects the disease, because this is what makes us reassured before we inject our patient with some radioactivity. We want to make sure that it will really go to the cells that it wants to kill. And right now, we also have a good expansion of research, both in chemistry and radiochemistry, for a more accurate diagnosis of specific cancers, so that we really specifically look for special targets instead of generic targets for disease, so that we protect the healthy cells, and we only want to go and send the cells to apoptosis that are the malignant ones. And the estimations say that we are going to have an increase of about 25%, which is huge, by 2030, which is basically tomorrow. So obviously that is going to be a big demand. And this is exactly the problem, because if we have those diseases that are treated and patients talk among each other. And so they all want to go for something that is going to treat and have no side effects. So how do we do this? Well, we definitely do more research. That's one thing. But it's not only about understanding which tracer we can kind of qualify to go to target. We also have to have this radioisotope that goes together with the tracer. And here I am actually showing the number of trials per different receptors. All the little flowers here, it's like the petals are receptors, so targets, And here you are actually at commercial phase. Here you are at phase 3, phase 2, phase 1. So this is basically to tell you how populated this area of nuclear medicine research, both for therapeutic and diagnostic parts, is. And this is evolving very fast. I would actually be able to show you the same slide about 5 years ago. It was a total different scenario. And so obviously, if we have that many patients and that many targets, well, we also need some isotopes in bigger quantity and differentiation, because we are going to have some radioisotopes that are better suited for some diseases than others. Therefore, we need to have a sort of sampling of isotopes. And obviously, the point is, where do we get those isotopes? Well, right now, the global production is actually already insufficient. And so we sometimes, even 5 years ago, before we went to market and so on, sometimes we would actually not be able to inject our patients because we just didn't have isotopes coming, because some of the nuclear reactors that we supply from are aging. They are then needing more maintenance, and they are not always dedicated to medical isotope production. They are also dedicated to research, to other purposes. Therefore, we don't know exactly how we started saying, hey, we are going to be in trouble because we don't have enough material to create radioisotopes, or um, to bombard so as to have some parent compounds and so on. So this aging infrastructure was not only a problem for us, but also for the regulatory systems, because they said that some reactors are no longer um, I would say compliant, so as to produce what is eventually going to be used after filters for medical purpose. So we really had some disruptions, and that cannot be, because we need more and we actually have less. So that was a big issue. And in addition to this, well, we basically count on 6 nuclear reactors all over the world. And that's not enough, because that is so there are some areas in the world where you almost cannot reach with radioactivity, because the patients there are not going to be able to benefit from the same treatment as others, for example, in Europe or in the US. And that is very unfair, because the patient should not be disqualified just because he was born in a place where the technology maybe is not as advanced. So we also had these logistical issues, uh, obviously, on the top of the fact that sometimes it's not easy to work with radioisotopes, because, well, first of all, they decay. Sometimes they decay by emitting, I mean, by also producing impurities that we are not happy with. So we have to kind of get rid of them. So the way to filter is not always easy. And if that's not enough, not every country has the same regulatory aspects as far as radioactivity. So it also differs from where you are, and that makes even more complicated all the supply chain availability. So in in addition to all this, another part of the world has said, okay, there are so many more patients that the current PET device or SPECT device are going to take too long to actually acquire an image. So we are going to improve not only the quality of the image, but the time of the acquisition, so as to reduce it. So we have more patients who are going to benefit from this positron emission tomography. And by reducing the time, maybe we can of the acquisition, we can also improve the detection of the annihilation. Therefore, we have a better quality and maybe we can reduce the activity that we need to inject into the patient. And this is exactly what happened. We nowadays have, um, I would say, 5 or 6 new models. That is a little bit the first model you looked like in 2018, and it's from a company that is Chinese, United Imaging, and it's a very good machine. And it also got even better or even more improved, I would say, by the version in 2024. We here acquire images very fast with an extreme good quality, and both Siemens and GE all have all as well provided models with what is called whole-in-one or whole body PET or total body PET, which means you actually have not only a piece of your body that is under the detector, therefore you can acquire the counts coming out of it, but all in one. And that actually is something amazing, because by doing this, what happens is that you can link what is going on into your brain, to what's going on into your stomach, to what's going on into your right foot, if you want. So we are going to even have better data out of it, and it's maybe a way to better understand our body, how our health is dominated by the body and by the environment, and not only by our will. So basically, what is ongoing now is that we really have only 22,000 patients and 35,000 patients. But what is going to become is much more, because we have a sort of triple projection. So we are going to have so many more clinical trials, and the advanced in the diagnostics as well, by the reduction of the time of the acquisition and of the machines and the better quality of life, that we really need more supply. And we have these production constraints because the reactors are aging, and we cannot run that far away if we have a short-lived isotope. So what kind of solution do we have? Not much, because it seems like we are actually only going to damage our patients if we don't do anything about it, because we are not going to be able to provide what they would benefit from. So we thought about an idea of how we could answer this challenge and also make it more more homogeneous as far as accessibility, because obviously nuclear reactors are very expensive, and it also takes a coalition of maybe governments, or at least powers, to accept to build one. So by having small modular reactors, well, maybe this is an opportunity that can actually benefit some of our cases, because we could have more than one, and by having them spread here and there, we could then be closer to some areas where now it's totally impossible to get access to. And maybe it doesn't cost as much, although it's still not super cheap. And maybe we can also take advantage from the fact that we don't have to invest into something only for medical purposes, because small modular reactors are obviously also meant to provide energy. So what if we combine the isotope production together with the energy, and when we actually are not using the small modular reactor for energy purposes, we take the kind of leftover and we do what can help the traditional isotope production, so as to at least level up to the demand that currently is around. And that was basically something that started becoming more and more into people's mind. And there was a phase of conception, duality, is it feasible, and so on. And due to the constraints that were put on the table to governments and so on, then and due to the fact that the clinical demand was very important, um, well, there was a thought of implementing this technology of the small modular reactors in this dual possibility. Some are actually predisposed, maybe more than others, but I'm sure that it's an ongoing development and it will become, I would say, every day more suitable for both purposes. Where it is needed, obviously, because maybe we will have so many that it's not even going to be a necessity for all of them. And in addition to this, according to which region, we could even think of having some small modular reactors that would be more dedicated to production of certain radioisotopes that are better to treat some diseases for which incidence is actually higher in those areas instead of having to produce and then ship it somewhere else. The idea would kind of be having a sort of short um, short delivery, like very to reduce the supply chain by reducing the distance from the producer to the end user with limitations of purity and so on, because obviously uh, it this is the problem that happens in the fact that several countries do not share the same regulatory framework. Therefore, may even between neighbor countries, like if you look at France, does not have the same regulatory framework as Belgium or as Spain or as Italy. So it always makes it very complicated because you would think that Europe is one for all. No. And you would also think that patients are going to actually be considered the same, but no. So that is an additional problem. And obviously, every country wants to feel safe for their own non-patients and for their patients. So they really insist in having their rules applied. So it's taking some efforts. And in addition to the regulatory frame, that would also enable the fact that if one small nuclear reactor cannot provide such isotope for some hospital or clinics, then we would also want to imagine that the closest one to the one that cannot can actually back it up. So we would have a sort of redundance so as to optimize the supply chain and um, and reduce the delays for treating those patients. Do it or not, the tumor does not stop growing. So this is always our idea of let's try to be fast. And so this really is a sort of sustainable model because it combines healthcare and energy. And it's a little bit our vision in the future, so as to try to optimize what creates in one way energy and in another way energy of life, so as to make a reliable supply of isotopes and with a shorter distance to the patient. That would be the first point. The second, ideally, would be that we can harmonize the dosimetry so that all over the world, wherever we are, we actually have the same standards. And when I say something, it actually means the same as when somebody on the other side of the world says something, because sometimes even among people who work on the same subjects, there is a little bit of incomprehension, even like international system units are not always utilized. So sometimes it's a little awkward. And finally, it would be very interesting if we could have a digital tracking so that we know exactly where radioisotopes are going from A to B in in real time, so we kind of really know where the patient is and how to best supply some radioactivity for that kind of disease to that kind of patient who weighs that many kilos. That would be the super dream. And in a future outlook, what it all started a little bit with this getting to market of the Pluvicto, which really enhanced the prostate community. And since it's not a small disease, it really questioned how would we decide if we don't have enough lutetium that one patient can be treated and the other one cannot. Who are we to decide? We really have to make it available for everybody. And so in 2024, we really thought that it was going to be important to act on this lack of radioisotopes, because if all the ones that enter in clinical trials are going to make it to market, we are going to be not not ready to actually provide treatments to patients. And in fact, this is in 2026, and there is really one small modular reactor that has been tested for production of energy and medical isotopes. So it did produce actinium. It's not going to be very popular because it's only one, but we actually are very happy about having seen that it is feasible. Now, the idea is to have many of them and very cheap and very standard so that everybody can use the same supplies. That is not exactly going to be easy, but in our mind, we would love to have by 2030 at least like maybe a third of the production of the medical isotopes that comes from small modular reactors. Now, maybe it's utopic, but this is what we hope for. And that would enable the fact that the radiotheranostics can have a supply chain that is really redundant enough so as to never break down, which is super important, because once we secure the supply chain, we can treat the patients. And if we scale precision by scaling precision theranostics through this innovative isotope production by the small modular reactors, we would really like to meet the growing demand in nuclear medicine, and we continue really believing that it is important to address those questions as early on as possible, and fostering collaboration across sectors with energy, with so many others, and implementing these advanced technologies, we can ensure the sustainable, equitable supply of radiopharmaceuticals, eventually to treat our patients, probably mostly in oncology, but why not in other non-oncological diseases as well? So on this, I wanted to thank you for your attention and hoping that altogether we can make a better world, a better impact for Mother Earth and for our patients. And I thank you very much and happy to take questions.
Thank you. Thank you very much, Laura. It was really quite a dense presentation, and I can see that there are already a couple of questions, and I have a few questions myself. And so let's start with the question that I have here, and then maybe I move on to the one I have that might be going to a few points, in fact. So let me start here. So Ravi Menon is asking if after the theranostic process, how is the curative process executed? Are they delivered through immunoassays using nanoparticles?
Okay, so yes and no, but it could be that it is a nanoparticle, but most of times it's not. Actually, the lutetium, for example, is in a chelator. Usually it's a DOTA chelator or something that actually chelates the gallium or any metallic. So it could be lutetium, it can be a nogada, and it is linked to the tracer, and by injecting into the patient, it goes directly to the tumor or to the cell that express that target. And that's it, basically. Then the irradiation I mean, basically the decay happens so that you have the radiation going either penetrating a couple of microns or according whether it's an alpha or a beta. And then you have so much energy that reaches usually the DNA of the nucleus that you have a sort of instability. It breaks the bonds between the two strands of the DNA. And
Yes.
once it's broken, then the cell goes to apoptosis. So it is a way for the cell to say, you die now because you're not perfect anymore. So it
Right.
dies. And
And so,
this is,
so it's not a nanoparticle, but in fact, it's another carrier, which is the metal.
yes, it's a chelator usually. Now, there are studies with nanoparticles, and those nanoparticles would actually totally go around the isotope. So even if it decays, whatever decays, daughters and so on, will stay in that nanoparticle. So in a way, it's very nice because since it goes through the blood, then it flows all over our body and it will, yes, decay. So it will release energy, but mostly uh, by going by, it only affects very little until it gets to where the target is, and there it will release because it will stay, you know. So this is a way to usually protect our healthy cells from critical irradiations that are not supposed to happen.
Right. And so let's say the beauty of those type of radio tracers is that they are really organ dependent. So they are really targeted for a specific organ. So they go
Yes,
there and they are not really targeting any other part of the body.
they distribute on, exactly. Yes. And this
Right.
is why it is so specific.
And so and so powerful. So the
Yes.
next question is, how long does it take for the isotopes to leave the body? Does the waste present an environmental problem?
Yes and no. So very good question. So obviously you have to get excreted, you have, well, the biological half-life and the physical half-life. So let's say if you take gallium or fluoride, then you have either 1 hour or 2 hours. So basically 10 hours after the half-life, you basically have nothing. Okay, so for those imaging radioisotopes, which we consider the day after, you have no more radioactivity in your body. But in between, what happens is that you probably have gone to the bathroom, and all those imaging tracers have a kidney metabolism. So they will actually be excreted through your kidneys. Therefore, we usually ask our patients to drink quite a few glasses of water before getting imaged. Then they get injected, then they get imaged, and then they basically go to the bathroom, and their urine is indeed radioactive, but this is already a big chunk of the radioactivity that is going to leave their body. So the patient actually gets very little uh, in his body or her body besides what is going and targeting, because if it keeps circulating, it will actually be excreted. So this is the beauty of it. If it
Right.
doesn't find anything, it goes through the kidney and then it goes into the bladder. And we indeed ask the patient to go and urinate because we don't want the bladder to get irradiated, the wall of the bladder. We don't want it to hold, you know.
Course.
So
No, but I've seen that there are special collections of the urines
exactly,
in such a way that it is not really released into the environment. So it would not really go through the normal sewage system. It would just be
exactly.
collected and kept until, of course, the half-life is such that it is not contaminating the environment. I think this was the point, that it is not,
Yeah,
in fact, a waste problem, because I think the hospitals, they have systems to preserve and
exactly.
keep, right?
In fact, when the half-life is longer for therapeutic purposes, indeed, we ask the same thing to the patient so that we actually try to free as much as possible his body from the radioactivity, except the one that is targeted, so is bound. But the bathrooms are special bathrooms because indeed there is a collection of the radioisotopes so as to wait until the decay is low enough, I mean, has happened enough so that the radioactivity, the activity is minimal and it can be released. But that takes with cubes and so on, so as that we make sure that we do not irradiate the environment because of the patients that are treated.
Thank you. So the next question is on artificial intelligence. Has AI been used for data collection analysis and critical decision implementation for isotope development?
So yes, it has been. It's not probably it was probably not one of the first subjects that have been taught to AI. So it's still ongoing. Sometimes it doesn't enter very accurately. So we are also training, but AI is a beautiful way to think wider, but at the same time, it can only think according to what you have taught it to think. So right now, it's almost like we have to uh, ask everybody to brainstorm with us so as to have more ideas coming because we can accept everything, but sometimes we don't know exactly what would actually be the best. So we do use AI to collect a lot of data, but we cannot always ask AI to think out of the box, I would say.
Okay. So you mentioned regulation standards, digital tracking, and well, they seem really to be linked together. So my question here is, first of all, who is deciding, and how long do you think it will take?
Okay, so who is deciding? Usually, everything that was radio related came from external beam radiation therapy, and those all the radiologists made the rules because that's what they knew. They actually worked with X-rays and so on, and they built a sort of threshold for each of our organs according to um, how much uh, uh, could be received for example, for the kidney, how much could actually go through the kidney before we would get a kidney failure. Okay, so for example, and I'm talking about the kidney, but it's a true example. There is a threshold of 23 grays that you cannot get to the kidney. Otherwise, I mean, you cannot go above that threshold. Otherwise, you're going to most likely induce a kidney failure to the patient. Okay. Now, we personally have treated patients up to 49 grays, and the patients are happy and they are very fine. Okay. So, what we tried to explain to the regulations is that the way radioactivity works, or at least absorption of radioactivity works when it goes from outside to inside, so such as external beam radiation therapy, is totally different than when it comes from inside because we have different cells that are differently radiosensitive or radio resistant. And for example, when you come from outside and with a beam, then you basically have to go through your abdomen and your a little bit of blood and a little bit of skin, and then you get to kidney. Now, when you get to the kidney through injection, it means that you are through the blood and then you go through the glomerular part of the kidney. which is very much more radio resistant. So this reuptake does not prevent the kidney from becoming toxic, or I would say it's not a problem for the kidney to actually have more radioactivity than 23 grays. But this is something that the regulatory bodies have a little bit of insecurity with, because they only heard of internal radioactivity from very recent, from 2017, as opposed to X-rays that actually it's been 100 years. So obviously it's going to take some time. Now, what we have succeeded to do is when we do clinical trials, we can go up to 29 grays, and they agreed. But we don't know yet as far as acceptance to commercialization, if they will. And it really depends on human decision. So if we have somebody saying no, it will be a no, regardless from the scientific proof and evidence.
Right. No, what I was even thinking is that so small modular reactors are now being built by private companies, and they are going to be sold. They're going to be used even well in the microreactor format, even for vessels for transport. And
Yeah.
therefore, these regulations they really need to be across the board. And
Yes.
so I would imagine that the same type of regulations that will be affecting transporting and using a reactor being built in one country to another country will be affecting probably also the production and the use of the radioisotope. So I think it's probably quite an interesting point that everyone would have to think about. So I think we are really getting closer, and there is another very technical question here that I'd like to bring to you as a last question. So, production of radionuclide requires frequent fuel removal at specific items, and SMRs are not designed for frequent fuel replacement. How is this conflict to be resolved?
Very good point. So at first, that was obviously a problem, because we thought, well, how do we want to change to put our target there if we cannot? And how do we want to remove it if we cannot? So lately, I think it's called a rabbit or something like this. They have a bizarre name of a sort of tube that goes in and I would say is blown, because it's like pushed by air to a sort of critical point in the reactor, so that it gets irradiated for the amount of time that you decide for that to stay, and then it gets um, like evacuated by vacuum again. So it's this, and that's why it's called rabbit, because it seems like it's running in and running out, uh, and it will then be used. But obviously, it's all automatic, and it's a sort of pneumatic tube, and it is not something that a human would ever do because of the irradiation things. But it's something that has been prototyped and I would say not used in clinical, obviously, but tested already because it was already in 2025. So that is where we are. But it's a problem that has been faced and hopefully a little bit solved. But thank you for the question.
So technology is really thinking of it because, of course, it is too important, and I'm sure that technologists and scientists and us will really do what is necessary to make sure that problems like this one are going to be solved. And so I would like to conclude by a comment that was posted on the chat for you, which from Michael Williams saying thank you for this amazing presentation. It provides much hope for this technology to save lives in case these patients might not have other options. And with this note, I really would like to thank you, Laura, for indeed this amazing presentation. you're really bringing a lot of hope. Some people in the chat, maybe they were expressing a little worried because, of course, we're really dealing with very serious, problems and of course the solution needs really to be at the same level, but there are really very high hopes that this would become another important way to save lives. Thank you very much, and thanks everyone for attending and for being with us until the very last moment. This presentation is recorded and therefore will be available for further views. So the next presentation will be the 18th of February, again, in applications and some spin-off of what we are now using very importantly for fusion. So will be plasma, the topic again. And so I hope to see many of you present also for that very important application. Laura, thank you. Thank you very much.
Thanks.